all been affected by the coronavirus pandemic, and so far the vaccines have been helping prevent the spread. But the threat of mutations still looms over our heads, as it does with any virus such as HIV or influenza. But how do viruses mutate? To understand where mutations occur, we must understand where viruses multiply. This is a cell, and it's needed for the virus to multiply, because the virus has no organelles. Instead, it's just a protein coat around some genetic information. The virus will use these organelles in order to help synthesize proteins and RNA. First, a virus must bind to the outside of a cell. Through endocytosis, the cell will engulf the virus. Then the virus will travel to the nucleus and attach, where it can then put in its genetic information. At this point, the cell is practically a slave to the virus. It now is producing only products to help the virus reproduce, creating proteins and replicating its DNA. Antigenetic drift is a kind of mutation in which small little mutations accumulate. They're typically mistakes in RNA or DNA replication. This is more common in RNA viruses than DNA because RNA polymerase lacks the ability to fix mistakes in the new strand. Can you find the difference between the original strand and the mutation strand? One single change in the nucleotides, as shown here, can create a change in the protein structure. This can change the shape of the antigen on the virus and cause the cell not to be able to have the antibodies to bond to it correctly. It's like trying to fit the wrong shaped block into a baby toy. It's impossible! Antigenic shift is a sudden change and requires two viruses. There are two types of antigenic shift, recombination and reassortment. Recombination occurs when two viruses with single-strand genomes end up infecting the same host cell. Then the strands will be stitched together to create the new virus's genome. This creates a scary Frankenstein-type virus. Reassortment requires two viruses with multi-stranded genomes to infect the same cell. Then, a few strands from each cell will be selected and put into the new virus's genome. This would be like taking the 4 strands of RNA from virus 1 and the 4 strands of RNA from virus 2 and shuffling it together. Then taking the top 4 cards, and this would be the new virus's genome. So we have all these crazy types of mutations, but how do they affect us and the rest of the world? Well, natural selection will act upon these mutations to select the ones that evade our vaccines. This means that the dominant strain will become the virus that is resistant to this vaccine. And this forces scientists to have to create yet another vaccine. Thank you so much for watching!